Hello everyone. In our today's lecture of Genetics and Molecular Biology part first, we are going to learn about the linkage and the crossing over. Now, before moving towards the point, one should understand what is the linkage and the crossing over. When two or more genes resides on the same chromosome, they are said to be linked and their transmission pattern is called as linkage. Here the genes uh, far apart on the same assort independently are not linked properly. These linkage they may be linked together on one of the connect autosome or connected together on the sex chromosome, or the genes on different non-homologous chromosomes are distributed into gametes independently on the another one. Genes on the same chromosome, however, tends to stay together and during the formation of the gametes. It generally occurs in the prophase of the meiosis first, where the homologous chromosome break at identical location and the rejoining with each other is carried out with the help of linkages. It is used as a mapping tool and genomic studies, and it was this study was first carried out in the Drosophila. All the genes present on a particular chromosome forms a linkage group, and the number of linkage group of a species correspond to the number of chromosome of that species. For example, in human beings, if we consider about the human male, 22 pairs of autosomes plus one X chromosome and one Y chromosome, that is 24 linkage groups are present. And in female, 22 pairs of autosome plus 2 X chromosome, that is 23 linkage groups are present in the human beings, whereas in Drosophila, 4 pair of linkage groups are generally present. Then the gene mapping. Now, gene mapping is nothing but the linkage of a genes in a chromosome can be represented in the form of genetic map or linkage map, or it is also called as chromosomal map. The first individual to construct the genetic map was scientist Alfred Strutwans. He used the frequency of crossing over between two genes to produce the first genetic map of X chromosome of Drosophila. There are two major aspects of genetic mapping. First one is the determination of the linear order with which the genetic units are arranged with respect to the another one. Means here the linear order of genetic units are arranged with respect to one another, and second one is the determination of relative distance between the genetic unit. What is the actual distance between the genetic unit that is determined? Now there are two types of linkages present. First one is the complete linkage. The complete linkage means the genes located on the same chromosome remains together and do not exist independent assortment are known as complete linkages. Here they do not get separated or they remain very close to each other. And incomplete linkage is means they can separate because of distance. When the distance between the two units is more, then they can separate from each other, and such type of linkage is known as incomplete linkages. Next important aspect is crossing over. Now crossing over is a recombination of genes due to exchange of genetic material between two homologous chromosomes. When in between two homologous chromosomes, for example, uh, capital T, when the uh, there is exchange of genetic material, exchange of genetic material is carried out in this homologous chromosome for the recombination of genes. It is known as crossing over. It is mutual exchange of the segments of genetic material between non-sister chromatids of homologous chromosome, and it produces the recombination or new combination of the genes. It occurs in the pectin stage at 4 strand stage with the help of enzymes. Different types of enzymes are used to carry out the crossing over, like endonuclease, exonuclease, R proteins, or recombinase enzymes are used for to carry out the crossing over. Now there are different types of crossing over. They may be single crossing over, in which the crossing over occurs at one point between two non-sister chromatids of a homologous chromosome pair. Generally, when only at single point, if the crossing over occurs, then it is known as single crossing over. Second type is the double crossing over. Now name itself indicate, now here the crossing over takes place at the 2 points of in a homologous pair of chromosome, so it is known as double crossing over. And there are two types of double crossing over. One is reciprocal and second one is complementary. Now the 2 points of crossing over when occurs between the same non-sister chromatids, that type of crossing over is known as reciprocal crossing or double crossing over. When same non-sister chromatids are present, and when at 2 points of same non-sister chromatids the crossing over occurs, such type of crossing over is known as reciprocal double crossing over. Second type is complementary crossing over. Now here generally it involves 3 or 4 chromatids, and as 3 or 4 chromatids are involved, the crossing over also takes place at 3 to 4 points. So the complementary crossing over is the two crossing over involve 3 or all the 4 chromatids, so that the number of crossing over is 3 or 4 with the occurrence. Then the next type of crossing over is multiple crossing over. Here 3 or more points of crossing over occurs in the same homologous chromosome, and double crossing over and the parental types may or may not occur here. Then there are different factors which influence the crossing over and the linkages. There are different types of factor which includes distance between the two genes, then the age of that person or the organism, then the sex, x-rays or the exposure to the various radiations, then various physical factors like temperature also affects the crossing over, and the most important factor is the contact with different types of chemicals. Now how does these factors influence the crossing over? Now distance, uh, when genes are at the closest location, there are the less chances of recombination, means nearer the genes lesser are the recombinations. Then the age, as the age increases there is a decrease in the degree of crossing over in the most of the species. Next factor which influences the sex. Now if we consider about the Drosophila species, male Drosophila shows little crossing over, and the phenomenon of crossing over is quite common in the female fly, or it may be negligible. Crossing over is also reported in one sex of the some other heterogenetic organisms. Now here the crossing over, here what happens, the negligible crossing over is occurred in few species or one of the heterogametic organism. Then the x-rays. Now exposure to such radiations or the x-rays increases the incidences of crossing over. Now temperature, as the temperature increases the frequency of the crossing over also increases, means the crossing over it completely it depends on the temperature present over there. And the chemicals, the number of chemicals present in the food have been found to change the degree of crossing over in the animals. Now the type of food they consume are also responsible for the crossing over occurred in that animal or the organism or human beings. Then what are the important or what is the importance of the crossing over for the diversity of biological evolution? Now due to crossing over, first important factor is new species can be produced, then mutation and the evolution can occur due to crossing over. From a single cell to a large animal or human beings, the variation can be studied with the help of crossing over, and a struggle for the existence can be improved by the help of crossing over. Now there are few important factors regarding crossing over and gene linkages one should take in consideration, that is crossing over is a means of introducing new combination of genes and hence traits, means crossing over, after carrying out crossing over we can introduce new type of genes or carry out different types of combination of genes in particular animal, so his characteristics can be changed, mutation can be caused, or new species can be produced by carrying out the crossing over. Second important factor is it increases the variability which is useful for the natural selection under the changed environment. Now as the environmental conditions are also changing and there is lot of struggle for the survival, struggle for the existence, for such purposes to survive in that conditions different types of variations are required in that animal which can be carried out with the help of crossing over. Since the frequency of crossing over depends upon the distance between the two genes, the phenomenon is used for preparing linkages and chromosome map. It is proved that genes lie in a linear fashion in the chromosome, and useful recombinations produced by crossing over are picked up by the breeders to develop useful new variants of the crop plants and animals. Green revolution has been achieved in India due to the selective picking up of useful recombination. Operation Flood or White Revolution is also being carried out on the similar lines. Now, after carrying out crossing over, when it is carried out, the selective recombinations are picked up, and then the multiple copies of that recombinations are breeded so that environmentally, what we say, in natural selection also those species will be selected, and in adverse condition also that animals or plants can survive and give the larger yield. Such type of crops or animals are produced. These are the important factors related to crossing over. Thank you.